Hello, welcome to Biology for You. In this video, we are going to discuss yeast-based vectors. In the series of videos, we have been discussing different processes in genetic engineering. So, as a part of different molecular cloning systems, here we are going to discuss these yeast-based vectors. So in this video we are going to discuss the basic idea of the yeast vectors. That means what is the yeast vectors and what are yeast vectors and what are the different types of yeast vectors and what are the basic differences between these types of yeast vectors. So coming to the first point, these yeast vectors are kind of molecular vectors. This molecular vector is nothing but a carrier DNA molecule which carries the target portion of DNA or a target gene to the host organism that is going to be genetically modified. So that means it just transport or carries the target DNA to the host organism. So once after it carries to the host organism will start expressing that gene or it start expressing or multiplying. Even in the case of molecular cloning, it start multiplying this target portion of DNA, and due to which it is now considered as a genetically modified organism or transgenic animal or plant. So that is what the molecular vectors are. So there are many kinds of molecular vectors, like very popular examples are plasmids, cosmids, phosmids and all. But all these examples are generally to clone DNA inside the prokaryotes like bacteria. But what if we want to clone some genes in order to inside the eukaryotic system? We need to have such vectors which are stably maintained inside the eukaryotes, because if you by doing molecular cloning inside the eukaryote, these genes which are eukaryotic genes which are more complex than the prokaryotic genes, and they need regulation system that is different from the prokaryotic system, and these eukaryotic genes generally need post-transcriptional and translational modification to be expressed properly. So for all these problems to encounter, we need to have a eukaryotic system, and such as yeast cell which is able to easily manipulate and which can be easily studied, which can be easily cloned, which can be easily multiplied inside a lab. So we need to have such system, and also we need to have vector that can be easily cloned inside such simple experimental systems such as yeast cell. So what if we modify yeast plasmids only in order to clone our target gene? So that is going to be best choice for the eukaryotic gene expression or eukaryotic gene cloning. So for that, these yeast vectors are quite helpful. So these yeast plasmids are basically covalently closed circular DNA, extra chromosomal DNA. They have self-replicating ability and all. So there are many kinds of, basically these yeast cells, why they are experimental choice means they are simple organisms. Yeasts are very much simple, very less complex organisms, and these are small, having haploid life cycle, and their lifespan is very much small. So due to the short lifespan, we can easily clone and we can easily multiply a yeast cell, and we can easily propagate, we can easily maintain a culture in the labs. So, and also these yeast cells are basically widely studied for their physiological and genetical characters. So all these properties makes the yeast cell suitable for the molecular cloning experiments for eukaryotic genes. And also this is a eukaryotic system, so it is having post-transcriptional, it provides this post-transcriptional and post-translational modifications inside it. So this is the best choice for the eukaryotic gene cloning experiment. So these yeast vectors, basically there are 5 types of yeast vectors. So for design, and these are, I better write it here. So first type of yeast vector is yeast integrative plasmid, and second one is yeast episomal plasmid, yeast replicative plasmid, yeast centromeric plasmid, yeast artificial chromosome. So these are at least 5 types of yeast derived vectors which are modified from yeast plasmids. And this yeast integrative plasmid is basically okay, I'll draw a rough diagram over here showing the important regions inside one such plasmids. So let us consider it as a yeast integrative plasmid. So this is the first basic type of yeast based vector. So it is basically bacterial. It is basically designed to be cloned inside the bacteria only, but it is having the ability. These are basically all these are basically shuttle vectors. That means we can clone inside the bacteria as well as inside the yeast cell. So these are shuttle vectors, and all these are shuttle vectors. So as yeast integrative plasmid as well. So this yeast integrative plasmid have origin of replication, that is the region where the prokaryotic gene replication will start. So this origin of replication or origin is taken from the Cole-Evan plasmid. So this is useful for the replication of this such plasmid inside the E. coli cell. So these yeast integrative plasmids are able to replicate inside the bacteria, and so these are also have selectable markers like amphicerin resistance to select them inside the E. coli cell, and these are not having the self-replicating ability once they introduced into the yeast cell, but they need to integrate inside the genomic DNA to replicate. So hence they are called as yeast integrative plasmids. They don't have the origin of replication for the eukaryotic replication. So they need to integrate inside the genomic DNA of the yeast cell. One such example of yeast integrative plasmid is YIP5. So coming to the yeast episomal plasmids, these are also the similar vectors which can be replicated inside the bacteria and inside the yeast cell as well. And these are episomal plasmids. That means these are kind of extra chromosomal elements. They have the self-replicating ability. They need not to integrate inside the genomic DNA always for the replication. Hence, these are called as double shuttle vectors. These yeast episomal plasmids are basically double shuttle vectors because they can replicate inside the bacteria, they can replicate inside the yeast nucleus by integrating into the genomic DNA. They can also have the self-replicating ability as a plasmid. So, due to which these are called as yeast episomal plasmids. One such example of yeast episomal plasmid is YEP13. So coming to the next type of yeast-based plasmids are yeast replicative plasmid. As the name indicates, these are replicative plasmids. That means they have origin of replication for the replication inside the yeast cell. That means eukaryotic origin of replication, that is autonomously replicating substance or ARS gene will be present inside the yeast replicative plasmids. So they are basically, they can stably replicate inside the yeast cell, and they need not to integrate. They do not integrate inside the genomic DNA for their replication, because they have the ARS, or else they have the self-replicating ability. One such example of the yeast replicative plasmids are And coming to the next type of plasmid, that is yeast centromeric plasmid. This yeast centromeric plasmid is basically a type of yeast based vector which is having self-replicating ability due to presence of ARS, and it is having a region called centromere. So due to the presence of centromere, they are mitotically stable. So going back to the YRPR yeast replicative plasmids, these are not having the ability to segregate during the mitosis. That means if these yeast replicative plasmids are to be segregated from the mother cell to daughter cell, during this process of cell division, they eventually lost. They cannot segregate due to the absence of centromeric regions which need to be catched by these microfibrils during the cell division. So they don't have centromeric regions to be identified as a chromosomes to segregate from parental generation to the daughter's generation. So they will be eventually lost. But due to the presence of centromere inside the yeast centromeric plasmid, they have ability to segregate during the mitosis. Basically, these replicative plasmids as well as these centromeric plasmids both are having high transformation ability, but the transformants are not stable. So these are having the centromeric region and they can mitotically stably segregate, but they don't have the stability over the period of time, or they may eventually lost as a plasmids. So one such example of yeast centromeric plasmid is YCP50. So this YCP50 is basically a yeast centromeric plasmid, but it does not have the stable maintenance throughout the life cycle. So yeast artificial chromosomes are advanced version of all kinds of yeast based vectors. These are having the ability to self-replicate due to the presence of ARS. They have this mitotic stability due to the presence of centromere, and they have the selectable markers for eukaryotic selection. That means inside the yeast cell they can be selected due to the presence of selectable markers suitable for yeast system, and they also have these selectable markers for bacteria. They have origin of replication to replicate inside the bacteria due to the presence of gene called origin, and also they have the regions called telomeres. So these telomeres are the regions which linearize this plasmid gene. So they can be stably maintained throughout the life cycle of the yeast cell. So due to the presence of telomeric regions inside the yeast artificial chromosomes along with all other regions that are taken from the types of plasmids before we talk, and these artificial chromosomes are having these telomeric regions as well. These telomeric regions are stable ends, gives the stable ends which cannot be easily degenerated, or they protect the DNA, linear form of the DNA, and they allow the DNA stably maintained inside the inside the eukaryotic system. So these yeast artificial chromosomes are having telomeric regions. So as we discussed, all these vectors are having the ability to replicate inside the bacteria, inside the yeast. Some or the other way they can also replicate inside the yeast. So these all yeast based vectors are called as shuttle vectors, and also these are having selectable markers for the bacteria and selectable markers for the eukaryotic systems such as yeast. So these are the basic characters and differences between the different types of yeast based vectors. So these yeast artificial chromosomes are the basically advanced type of yeast based vectors which can really clone very large fragments of DNA such as 100 kilobase pairs to 300 kilobase pairs or such like that.